Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Let me introduce myself. My name is Efrin Firmansyah. I'm from Agrotechnology of UIN Sunan Gunung Jati Bandung. Today, I would like to share about our research about fruit fly attack on intercropping plants. As an introduction, as we know that fruit fly is one of the important pests that attack horticultural community in Indonesia. Red cherry or Capsicum annum is one of the hosts of fruit flies that is widely cultivated by farmers in Indonesia. In addition to red chili plants, fruit flies are also known to attack other plants such as orange, mango, guava, and papaya. The type of species in these plants vary from one another. The species include Bactrocera dorsalis, Bactrocera carambole, Bactrocera papaya, Bactrocera umbrosa, Bactrocera cucurbite, and Bactrocera alistrigata, which attack various plants and in different places. Fruit fly population can increase when environmental conditions are favorable, such as high humidity, low wind speeds, and moderate rainfall. The purpose of this research is to study the attacks of fruit fly in the intercropping plants between Capsicum annum and Cosmos caudatus. The method this research was conducted in a red chili plantation area located in Sukarara village, Garut, West Java, and the identification of fruit fly species was carried out at the FAST Laboratory, Department of Agrotechnology, Faculty of Science and Technology, UIN Sunan Gunung Jati Bandung. The trap used to determine and monitor the fruit flies population from the field is a slightly modified standard trap. And to identification of fruit fly species, we use the 200 samples, and the morphology of wings, thorax, and abdomen is the reference used for identification. The trap fruit fly samples were brought to the laboratory to do seen for morphology using a microscope. The results are used for species identification by matching the morphological character obtained from the laboratory with the morphological characters in the software. The final fruit fly identification results will appear automatically when the match is completed. And the result of species identification showed that there was only one species of fruit fly that attacked intercropping red chili and canicule planted in Sukarara village. Identification based on the main morphology of fruit flies, namely thorax, abdomen, and wings. The results of morphological observation in the laboratory that have been matched with the morphological sketch contained in the data in case. So that the identified species is Bactrocera dorsalis. This is the picture, morphological picture from the laboratory, and compared with the morphological sketch in software. The number of white lines on the thorax, there are two numbers, and there are T-shaped line on the abdomen, and the last wing venation. The thorax of Bactrocera dorsalis has a dominant black color. There are two bands, or pale white to yellow, as much as two bands on the right and the left sides of the thorax. On the abdomen, there is a line with a firm or clear T pattern starting at the third segment of the abdomen. Bactrocera dorsalis wing venation has no transverse band from the costal margin to the underside of the wing. The results of observation of the abundance of fruit fly population based on trapping results show that the number of fruit fly population fluctuated every time the observations were made. The lowest population was obtained at the time of the 60th observation, which was 916 individuals of fruit fly. Meanwhile, the highest population was found in the sixth observation, which was 3,258 individuals. Several things that can affect the size of the fruit fly population in the field include the individual distribution activity, the birth rate of new individuals, the death rates, and agroecosystem conditions that support the abundance of resources needed by fruit fly. These are also factors that affect the abundance of fruit fly population on agricultural lands. The conclusion is the first is the identified fruit fly species is Bactroceratha dorsalis attacked in intercropping plants. And second, the abundance of fruit fly in the intercropping area of chili and kanikir plantation fluctuated during observation. I think that's it for me. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.